So in this slide we see a picture of how Mendel did his cross. He tested lots of different traits. In this picture they're testing pea shape, so round and wrinkled. The way he controlled the crosses was that he would cut off the anthers, which is where the pollen forms. He would cut the anthers off before they were fully developed so that a certain flower wouldn't produce pollen. And then he could dust the pollen from another plant directly onto the stigma of the first plant, and that's the stigma leads down to, there's a channel that goes to the ovary. And so that's how he would pollinate, and which would lead to fertilization of the eggs. Now the ovary, which is shown here in a plant, contains the eggs, and the pollen will travel down the style, which is this part here, and it will reach the ovary, and the ovary will enlarge, and each of the fertilized eggs will develop into seeds, and the petals fall off, and so the ovary becomes the pod for the pea plant. And then the seeds can be planted and grown, and then we can see what the offspring look like. Okay, so that's the basic strategy for Mendel. He had to do a lot of peas to get the data that he got. Now when Mendel did, for example, one of his crosses, he used round seeds and wrinkled seed plants. Now he didn't cross the seeds. Of course, they're just showing the seeds here to represent the whole plant. So a plant that produces round seeds is crossed to a plant that produces wrinkled seeds. And so you do cross the pollen from one plant to the flower of the stigma style ovary of the other. Okay. Now when they say homozygous round, what that means is when they self-fertilize the round plant, the plant that makes the round seeds. Many times when they self-fertilize it, it will always have offspring that also have round seeds. So Mendel had to verify this. And then when you have a plant that's homozygous for wrinkled seeds, that meant that Mendel had to self-fertilize it many times to ensure that it always would produce offspring that had wrinkled seeds. So he had to pre-test all of these plants first, and then he set up the cross between one plant that was a round seed type plant and one plant that's a wrinkled seed type plant. And when he did the cross in the first generation, in the offspring of the first cross, he got all, in this case, round seed plants. So he thought that was interesting because he probably expected a blending effect, but he didn't see that. And then he would grow those plants up and self-fertilize them, and then when he got the next generation, he would look at what kind of seeds they make, and they make some round and some wrinkled. So, and he found when he repeated this with lots and lots of plants and counted up all the round seeds and all the wrinkled seeds, he got a 3-to-1 ratio of round to wrinkled. His actual data is shown here on the left side: 5474 round seeds, 1850 wrinkled seeds, and you remember, if you divide by the smaller number, you'll get the ratio, the observed ratio. And all of his observed ratios were coming out very close to a 3-to-1 ratio, no matter which trait he was testing on lots of different experiments. So this led Mendel to form his first law, the Law of Segregation. And segregation just means really separation of alleles during meiosis, specifically anaphase 1. But basically what his law says is that each diploid organism, or what we now call diploid, has 2 alleles for each gene. So you have, in a pea plant, you have a gene for pea shape, and each cell in each plant has 2 copies of that gene. But the alleles can be the same type or they can be different. One allele can be dominant, one is a recessive. A dominant allele means you only have to have one of the dominant type of allele in a cell in order for that dominant trait to be shown. The recessive allele, you would have to have 2 copies of a recessive allele in the cell in order for that phenotype to be shown. But when a cell goes through meiosis, especially a cell that might be heterozygous, having one of each type of allele. When that cell goes through meiosis to make gametes, half the gametes will get one of the alleles, maybe the dominant allele. The other half of the gametes will get the recessive allele. So in the mix of gametes, you would expect half to be represented by a dominant allele, and the other half would have the recessive. And then at the moment of fertilization, the new organism is restored to the diploid state when the two gametes come together. So Mendel came up with all this. I know it seems really basic now, but Mendel came up with this whole concept, not knowing anything about cell division, meiosis, chromosomes, diploid, any of that. He didn't use the word diploid, but he said that every organism would have 2 copies of each gene. He didn't use the word gene, but that's what he was talking about. And so it's really impressive that he came up with all of this. Now, how does this connect to the way the chromosomes behave during meiosis? From a meiotic standpoint, the law of segregation is really talking about how the homologous chromosomes are separated. And the homologous chromosomes are primarily separated in anaphase 1. Okay, that's the moment when the 2 homologous chromosomes are separated or pulled apart from each other. And then in telophase 1, that finishes the process. So if you have a big R, little r cell, then by the end of meiosis 1, half would be big R and the other half would be little r. And then meiosis 2 just makes clones of that. So for the most part, we're talking about an anaphase 1 being the key defining event of segregation. The only exception to that is if there is a crossover involving that gene, then technically the moment of segregation would be found in anaphase 2, as you see here. Okay, but as a rule, we're going to say that anaphase 1, the pulling apart of the homologous chromosomes, really defines, for the most part, the separation of the 2 different allele types. And so, once we know the law of segregation, we can actually predict what the gamete genotypes would be, as long as we know the genotype of the parent. So in this case, we have, we're going to use a Punnett Square, which I call a virtual fertilization on paper. I made that up. And what it is you take, if you know the genotype of your 2 parents, let's say you have a tall plant, but you know it's a heterozygous tall, and you have a short plant, but you know it's homozygous short, then what you're going to do is you look at that genotype and you say, okay, for this big T, little t plant, the law of segregation says half the gametes will be big T and half will carry the little t. So I represent that here on my Punnett Square. Half the gametes have a big T, half have a little t. For the short plant, since both of the alleles are the same, little t, then you can put a little t here, and if you want to, you can put another one here. You could really just put 1, but you can put it twice. Then when we bring the 2 gametes together in a fertilization that happens inside the box, inside the grid, then that shows what the genotype of the offspring would look like. So big T, little t would be a tall plant, big T, little t would be a tall plant, little t would be a tall plant, little t would be a short plant, and little t would be a short plant. So 2 tall and 2 short, a 2 tall to 2 short ratio, or a 1 tall to 1 short ratio, if you reduce it. And so the Punnett Square is really the way of just solving or predicting what the offspring ratios would be, as long as you know the genotypes of 2 parents. And Punnett was friends with some famous geneticists. William Bateson is the person who's credited with coming up with the word gene, and Godfrey Hardy was involved with the Hardy-Weinberg equation for population genetics. So this is Punnett here. Okay, but he came up with this idea. So I'm going to end part 2 right here.